Hi, class. So, if you were not in our regular 90-minute class, or if you just need a little bit of review of what we did during our regular 90-minute class, I wanted to make sure that you had a place to go back, review the information, and also practice a little bit on some of the things that we did in class. So, today, we're going to talk about transcription and translation. What the heck is that? I'm going to tell you all about it. Here's what you should do. Open your set of the slides. They're posted in your Google Classroom, and follow along with me. Every once in a while in this video, it's going to pause and ask you a question. Answer straight into your slides. At the end of the course, or the end of this video, I want you to submit your slides, and that will tell me that I'm ready to go back in and give you some feedback. Okie dokie, artichokes. Let's get started. So let's get a little bit of a warm-up. I am going to have you pause right now. Open up first this video by Learn Genetics, just part of Utah's university system. You're going to open up that video. It's short. It's about a minute and a half. Watch the video to sort of remind yourself, what is DNA? How is it structured? And like, what the heck are these base pairs? This interactive tool is going to ask you to practice matching the base pairs. So of course, A's always go with T's, C's always go with G's. Go ahead and give it a little bit of a practice. As I said before, this is our slides for today, so make sure these are open. All right, today's objectives. Last week during class, our goal was to describe the structure or the way DNA is made and to understand the size of DNA. It is so small, it is literally inside of our cells. Today we're going to move on. We're going to talk about the processes or the step-by-step, like the different steps it takes that turn my genetic code, aka DNA, into my traits. For example, how does my DNA actually turn into my eye color? Okay. So let's just do a quick reminder. Complete this DNA strand. Go ahead. C matches with what? G. G matches with the C. A. Great. Awesome. And we can just finish this up. So this is if I wanted to put together both strands of the DNA, right? One side of my DNA strand would be CGACGTC, and the other side would match up perfectly with it, and it would have the complementary pair. Awesome. Okay, let's take a second to think about this sentence. DNA is the instructions for life. Totally, absolutely true. We know that. But if I'm a writer or a science writer or really any kind of writer, I really want to make sure that I'm going beyond just that simple sentence that DNA is the instructions for life. So let's think about how we can go a little further. We use words like because, but, or so as conjunctions. They put two different ideas together. Of course, those ideas are going to be connected, but they're connected in different ways. So because is going to tell me why. DNA is the instructions for life. Why? Okay. DNA is the instructions for life, but is a different conjunction. But is going to tell us, let's go in a different direction here. That might be true, but this other idea is also true. Or DNA is the instructions for life, so. So takes us a little bit deeper. What can we do with that information? All right, so take a second. I want you to match these together, and we'll think about which sentences make the most sense. So you've done your matching, and I'm going to explain the choices I made. DNA is the instructions for life because I want to use the sentence end that is going to give me the next step in the information. So for me, that's because it contains all of the information that gives us our traits, right? That's why it becomes the instructions for life. DNA is the instructions for life, but everyone's DNA is unique and different, right? So all life has DNA, but everyone's DNA is different. Awesome. So these are two sort of separate ideas that are connected. And finally, DNA is the instructions for life. So, what can I do with that information? That tells me that I can read someone's DNA and learn a lot about them. So these sentences, this is just a little bit of practice with writing. As you get into more and more writing about science, we're going to use this information, and it's, I think, going to be really useful. Okay, get up, move your body. We just did a little bit of a warm up. Maybe you've been sitting through first period. Now you're sitting in second period. Get up, move your body. When it's all unraveled, the DNA in one human cell is 6 feet long. So get up, move your body, find me something else in your house that is 6 feet long. Okay, I'm gonna find one myself. I found something. So I have these two rods. Each one is like 3 feet. So 1 foot or one rod, two rod together. That's about 6 feet. It's about a foot taller than me. That's a ton of DNA in your cells. Type in the chat, what else did you find that might be 6 feet long? All right. So, proteins are the basis for all of our traits. Every trait, like in the world, every trait in every single organism is expressed by proteins. So I want you to take a second. These are like crazy science words: keratin, rhodopsin, actin. I don't even know what these mean. Take a second, Google these and see what is the function of keratin. Which of these traits actually matches up with the function of these traits, okay? So take a second, Google each one and see if you can figure out how they match up. Here's what I discovered. Keratin is part of my hair. So follicle cells, the like little cells on your scalp, actually make keratin. That's their job. They make keratin and it builds basically my hair. Obviously, my keratin's a little short right now. I'm going to need to get my follicles building more keratin. Okay. Rhodopsin. Rhodopsin actually helps your eyes. Your eye cells make rhodopsin to help you see, and in particular, it helps you see in the dark. Actin is a protein that every single cell makes, and it helps keep it strong. All right, we got these all matched up. Looks good, looks awesome. How does your body actually turn DNA into a protein? It happens in 3 specific steps. So very first, we're going to do transcription. Transcription, it sounds like this big science word, but there's really good information inside the word transcription that's going to help us. Transcription is like transcribe. This is what you do when you have a thought in your head and then you write it down. Your thought can't just leave your head, right? You have to do something to make your thought either on paper or speaking it out loud. That's called transcription. And our cells do something really similar with DNA. Once we have our new transcribed information, then we're going to translate it. And this is exactly what you think. It's like translating to a new language. I'm going to take the language of before and turn it into a new one. And then finally, I'm going to do some protein folding, which is like folding a protein, really. I don't have a good metaphor for it. Get it? The meat's all folded in that sandwich. Meat's protein. All right. Shut up, Miss Daly. Okay. Independent work time. So let's learn about these steps. The first one is transcription. Your job is to watch this video. Again, it's short. And then we're going to answer the questions here. So pause yourself and go ahead, watch that video, watch the transcription video, and come back when it's done, and we'll go over these questions. Cool, you watched the video, you answered the questions. Let's double check. So inside which organelle in the cell does transcription occur? This happens in the nucleus. The nucleus is that center part of our cell that keeps our DNA really, really safe. Same deal. Your DNA, it's so important. It is so important. It cannot leave the nucleus. That would be so unsafe. So your cells do transcription. They make a copy, okay? So we have our two molecules. The first one is DNA, right? Our beautiful pink molecule. You can see it's kind of got that double helix. It's DNA. And then your cell is going to make a version of that called RNA. RNA is what is the result of transcription. So in this process, the RNA is a copy of your DNA. And then there's something unique about RNA, and that's it doesn't have any T's. There's no T's in RNA. Instead, U is going to match with A for RNA. Okay? Awesome. So let's practice. Remember, RNA has no T bases. So your job right now is to turn the DNA, the TAC, TTT, GCA, GGA, turn it into an RNA sequence. So I'm going to do that by matching the complementary pair, but anytime I want to write T, instead I'm going to write U. So TAC is going to be complementary with AUG. T again matches with A. A should match with T, but RNA has no T bases, so A instead will match with U. And then C's and G's are all the same. TTT is going to match with AAA. GCA, G will match with C, C will match with G, and A will be with U. It should be T, but in RNA, it's U. And then we'll do our last one. We should have C, C, U. Go ahead and do the next one on your own, and then I'll pop up the letters and you can check yourself. All right, let's check it. UGA, CCU, CGC, and of course, AUC. Awesome. All right, so this is really important. I'm actually not going to go over this. This is all up to you. If you see a star there, that means I'm going to assess this. I'm going to put a grade on this one. So take a second, use the information. You can go back through the slides. You can watch that video again and take a moment to drag and drop each of these green boxes into either DNA, RNA, or both. All right, I can't wait to come back and look at these and give you some feedback on them. Take a break if you need it, if you want to keep rolling, rock and roll. All right, let's do the second step. We talked about transcription. Now our second step is translation. Again, you're going to pause, watch this video, also like 2 minutes long, and then come back and we're going to answer these questions together. This is sort of the second step. So remember, at this point, we turned DNA into RNA. What are we going to do next? All right, you watched your video, you answered your questions. If you haven't, pause, go back, make sure you answer them, and now let's double check together. The purple or blue organelle, remember an organelle is just a part of a cell, is called a miniature factory. This is a ribosome. Ribosomes are so important. They actually are the things that build proteins. It's like this little amazing molecule organelle in all of your cells that's like reading RNA and building proteins. So that yellow molecule is RNA, same as in the transcription slide. And the red molecule that it's building is called an amino acid chain, which will become a protein. So this is just a chain of amino acids. The RNA is read 3 letters at a time. Okay, 3 letters at a time. That's why when we see that like CCG, AUG, that's 3 letters equals 1 amino acid. So let's look at this in a little more depth. Here is my organelle, and remind me, what is this organelle called? That's right, a ribosome. And it's reading a molecule. Which molecule is it reading? RNA, exactly, that's exactly right. Now, each of the 3-letter sections, so here's this RNA right here, AGU, it's going to match with another piece, and it's going to add together an amino acid. So these amino acids are coming together And then all together, this chain is going to become a protein. So all my amino acids connected up together eventually becomes a protein. So the last step of this is that the amino acid literally folds itself up into a protein. I want to give you some examples of what the amino acids are because this is really useful for learning how to do translation yourself. Now I have all of these little 3-letter guys, PHE, LEU, VAL. They're just like abbreviations for longer science words like phenylalanine, which I'm not going to write down every time I need to answer this. I'm just going to write PHE. So what does this look like? This chart shows us all the different possible combinations of RNA bases, and it tells us which one matches with which amino acid. There are 21 amino acids, and all your proteins are made of some combination of them. So if I have this big, long amino acid chain, it could be that all the blue ones are phenylphthalein. So when I read a UUU or a UUC, I'm going to add a phenylphthalein or just a PHE to my amino acid chain. Then, anytime I have a pink one, that could be leucine. And that means I read any of these bases, CUU, CUC, CUA, or CUG, could be some other ones. Anytime I read those, I'm going to add a leucine. And then finally, just another example, obviously there's more than 3, but VAL means valine, and that could be my orange ones. And anytime I read an RNA chain that says GUU or GUC or GUA or GUG, I'm going to use valine as my amino acid. So let's practice this together. Oh, actually, this is cool. This is what it actually looks like when your protein molecule folds together. You got this big, long amino acid chain, folds up, and once it's all folded up, that's a protein. Cool. Oh no, I gave you the answers. Oops. So let's look at this. This first step is transcription. So this is where I turn my DNA into RNA. And my second step is to match my RNA base, GCGU, to the correct version, the correct amino acid. So I'm looking at this right now. GCA is the DNA. I turned it into RNA by matching G with C, C with G, and A, instead of with T, I'm going to match it with U. Now I need to figure out which of the possible amino acids is going to match with CGU. So this chart is actually really handy. So it's going to tell me my first base is C. So I'm going to find my C. And my second base is G. I'm going to find my G, and I'm going to find where those intersect. So I'm going to follow my C's along until I end up with G's. Now I have my C's and G's. So this one ends in U. CGU corresponds to ARG. So I'm going to type ARG right down here. Same thing with the next one. AAA matches with, instead of TTT, it matches with UUU. So I'm going to go look again. First base is U. Second base is also U. UUU. Phenylalanine, right? We did that one already. UUU. PHE. TCT, the RNA version is AGA. So I'll look. First base A. Second base G. So I'm looking for my AGs. It's not AGU or AGC. It's AGA. So that's also going to be ARG. ARG. And then finally, UGC. U matches with G, and then UGC. It's going to be CYS. Okay? Awesome. So go ahead and check your understanding. Let's put these steps in order. Which one do we need to do first? So the first step is that transcribing, right? It's taking the information in your head and writing it down on paper, or taking the DNA in your nucleus and turning it into RNA. So I'm going to move my transcription. That's my first step. Now I have my RNA and I'm going to turn it into like a new language. Instead of being RNA and being bases, now I'm going to turn it into something brand new. That's translation, right? This is almost like a different language. And then finally, easy peasy, thing folds itself up and it becomes a molecule known as a protein. So this is called protein folding. Radical. Totally rad. Okay, y'all, so you have the job to finish these two slides. I believe they are slides 22 and 23. I want you to wrap those up. So the first step is to turn the DNA into RNA, and then use your practice to turn your RNA into your amino acid chain. And then last, you're going to answer this question. I will read it out loud one time, and then you go ahead and answer it. You also need to give me one piece of evidence for how you know your answer is correct. The instructions for the genetic traits of an organism are directly determined by A, the numbers of A, T, C, and G units in a sugar molecule. B, the sequence of bases in a DNA molecule. C, the length of a DNA molecule. Or D, the way the bases are paired in the two strands of a DNA molecule. All right. You're going to answer that question. You're going to get yourself organized. When you're done, submit your slides so that I know I can go in and give you some feedback. Thank you for coming and reviewing with me, or thank you for coming and getting the information if you missed it before. Totally appreciate you. You absolutely rock. And I hope that this was really helpful for you. Okay. Have a really great afternoon, everybody, or evening, or morning, or whatever it is that you're doing right now.